harnessing environmental molecular data for Earth systems discoveries. Just a few reminders before we get started. Please have your laptop out. We will be doing a Miro board activity towards the end of this session where it would be easier to participate on your laptop. And then also please remember to sign into the Google Notes document. So with that, I'm Samantha Miller. I'm a project manager at the Environmental Molecular Sciences Laboratory, and I work with the user program. And it's okay if you don't know what that is right now. But I'm gonna let the rest of the speakers come up and introduce themselves before we get started.
Hi everyone, I'm Sarah Lichty. I am also a project manager at EMSL.
Montana Smith, I'm a data scientist at EMSL, and I also work on the National Microbiome Data Collaborative.
And I'm Kelly Stratton. I'm a data scientist as well, and EMSL's data transformations lead.
Great, thanks everyone. So before we dive into the agenda, I wanted to get started with a quick poll. So if everyone could please use the QR code on the screen and submit your answers to the question, have you heard of the Environmental Molecular Sciences Laboratory, or EMSL? This is not a very anonymous poll. Don't worry, safe space. A lot of no's. or one, I don't know. That's okay. Even if you haven't heard of us, we hope that after this session, you'll have an idea of how EMSL might be able to help with your research or the work you do. Okay, so with that, we have three main goals for this session. One, to introduce EMSL to the ESIP community, including talking about some of the science we do and the resources that we offer. Two, learn about the ESIP community and what your interests are. And then three, identify synergies between EMSL and ESIP. So to meet those goals, here's a quick look at the agenda. We'll start with a brief presentation to introduce you to EMSL, and then we'll do a quick word cloud activity to sort of learn more about you and what data types you're interested in. And next, we'll have a deeper look at the resources in the computational sciences, and Kelly will share some examples of user research. And then hopefully this sort of sets the stage to do a Miro board activity where we're going to try to identify some of the overlapping areas between EMSL and the ESIP community. So with that, I'll turn it over to Sarah to give an introduction about EMSL.
Great, thanks Samantha. So EMSL, or the Environmental Molecular Sciences Lab, we're one of 3 user facilities stewarded by and supporting the Biological and Environmental Research Program in the Office of Science. And EMSL is really unique in that our scope spans almost the entire breadth of BER science focus. So we both study the role of molecular processes in controlling the function of biological and ecological systems. We're looking across spatial and temporal scales. So we do kind of everything from the single biomolecules all the way to the ecoregion scales. And this serves really as a key integrator across different programs and other user facilities within BER. So how does our user facility serve the scientific community? Because that's what we're here to do. Scientists from around the world can partner with us and access over 200 different instruments and associated expertise. They can do that for free if their research proposal is accepted through our peer review process. And we collaborate on projects with researchers from academia, other government labs, and also industry. And there's a lot of opportunities to work with us available kind of throughout the year through our proposal process. And so over the next couple slides, we're going to explore both EMSL's resources separated sort of by science area, while highlighting that really the value that EMSL provides is that we have an integration of capabilities across science areas. So you'll kind of see that come up again and again in this session is we have these different science areas, but the value is integrating them, kind of crossing those boundaries. So the first science area that we'll cover is the computing sciences. We can think of that being broken into two sub-areas. So Emsl has expertise in both data transformations, and we have Kelly here to talk more about that, and also systems modeling. And so that includes integrating sort of disparate data types and also using molecular simulations to better understand an environment or an organism. And really this slide is just a high-level overview of Emsl's computing expertise, and we'll show some specific examples of computing user projects in later slides so you can kind of better contextualize what that work might look like. Each of Emsl's science areas has a corresponding decadal goal, and we're going to just touch on each one to give you more of an idea of where Emsl's pushing those scientific boundaries, kind of what we're working to accomplish over time. And so we provided these little nifty QR codes on each of the slides, and you can go to that spot to see if you'd like to kind of read more in depth about those summaries and get a better idea of what really is being accomplished. But from a high level with Modeling, Integration, and Data Agents for Science, or MIDAS, we love our acronyms, this aims to identify high-value opportunities where agentic workflows in predictive modeling, data analytics, and integration can accelerate science by increasing efficiency for multidisciplinary research teams. And really what we're getting at is how to make science go faster, make it easier for our researchers, and really the goal of this one will make more sense once you've seen our other two science areas, because they really do kind of integrate together. And so we'll talk about that in the coming slides. The second science area that we're going to focus on is the biological sciences, and that's split into three sub-areas that really focuses on elucidating and harnessing the biochemical pathways. And we're looking at how gene function connects to phenotypic responses by understanding interactions within cells, within communities, and within environments. And there's a lot to look at on this slide, but really the main takeaway is we have a bunch of really fancy instruments that can help us see what we can't with just our eyes. And so we're looking at biomolecules such as metabolites, lipids, proteins, really the foundation of all living things that impact kind of how our environments function, how organisms function. And so we have a couple images here that maybe help to make the abstract a little bit more concrete. So the image that's on that far left is actually mass spec imaging. And so we can look, and in this example, it's a plant root that on one side is experiencing drought and the other side was well watered. We were able to look and see differences in metabolite concentration. And really the value here is being able to see the structure of that plant root. It's not something that we typically are able to see is where those metabolites are concentrated, and that might tell us something about how a plant is responding in the face of drought, which we know is only becoming more common. So that's our mass spec imaging. In the middle, we have an example of some of our really high-tech microscopy techniques. And again, that's allowing you to see that micro scale, at molecular scale. And this is actually our lattice light sheet, which provides a 3D image of live cells. So you're actually able to see cells moving and interacting over time. And in this image, it's actually showing it's kind of a pretty picture to show like chlorophyll, that's the red color, and then also the green is dyed little lipid droplets. So it just shows you, like, you can see kind of the orientation of things in space and getting that really high resolution look. And then on the right side here, we have an example of something that it's really complex and took me a long time to understand when I got to Emsl. We're actually looking at a cryo-electron microscopy technique that can image proteins and like how structurally how they're formed, and that can give you an idea of the function. And in this example, we're actually looking at a protein that plays a role in manganese biomineralization. So looking at kind of those really small kind of biomolecules, but also really complex, right, in the different folds, to understand how that connects to the environment and how things are cycling. The corresponding decadal goal is called DigiFEN, which is the Digital Phenome platform, and that is where users will generate a variety of different data types, kind of use those data. And so it's using a lot of the techniques on the past slide to parameterize computational simulation and modeling tools to predict the behavior or phenotypes of biological systems of interest. I know that sounds really broad. Everything that we do is related to our sponsor, and so there are some specific species that are of interest to BER, and this is a decadal goal that's kind of getting up and running in the near future. And so again, you can look at that QR code, get a better idea of what that might look like, but a lot of it is kind of up and coming and new. We're developing capabilities and using those techniques I showed in the last slide to kind of build out what this might look like and bridge across these different really specialized techniques to have a fuller picture of what are different communities of microbes doing in the environment, and what can that tell us kind of on the ecosystem scale. So we're always looking at bridging across those scales. Our last science area is the environmental sciences, and again, that splits into these kind of 3 sub-areas that focus on mechanistic and predictive understanding of environmental processes in both above and below ground systems, and within the atmosphere and their interfaces. So sort of a catch-all for a lot of different things, and it's near and dear to my heart as a soil scientist. And so this first area with the biogeochemical transformations, really we're looking at the movement of nutrients and chemical compounds. We know that links back to microbes and what they're doing, but really we're looking at kind of a higher level view and understanding, for example, this image is of our 21 Tesla FTICR, and that is able to identify thousands of molecules that might be present in natural organic matter. So really important for understanding how compounds from decomposing plants and microbial communities, what's actually in that mixture, and what does it mean for nutrient cycling in the environment. So there's only like 1 of 2, I think, in the world. So we've got really specialized instruments as well as staff to help you make sense of that data. We also have techniques that can look at rhizosphere functions, so kind of diving into sort of the black box of soil and roots and understanding kind of where different molecules are both interacting with the roots and the microbial communities there. And the example we have here is terraforms, which is a technique at EMSL where they actually create a microscale environment that mimics soil properties. So it's a more kind of a simplistic version of what's happening in soil, because we know it's really complex, it's always changing, and so this is a way to better visualize soil microbial and plant community dynamics. And then lastly, we'll just touch briefly on our terrestrial atmospheric processes, and that focuses on sort of the molecular transformations that happen in the atmosphere and how they connect back to terrestrial processes. And we have this nifty little graphic from our communications team, and that's showing how the instrumentation kind of within this sub-area has helped to understand factors impacting aerosol composition above agricultural fields. And so this hopefully gave you a better idea of all these different techniques and how they can be brought together to better understand an environment, because we realize that not just one technique will answer all your questions. We have to use these different techniques kind of in conjunction to better understand our environment. Lastly, the decadal goal for the environmental sciences is the Molecular Observation Network, or we call it MONET, and that's developing a continental scale database of standardized molecular and microstructural data, and that's to advance our understanding of soil ecosystems as well as terrestrial aerosol processes and rhizosphere. So this is actually a project that I manage, so I know the most about it, if you have questions about it. And we are going to dive a little bit more into what we're doing with MONET to engage with the scientific community and kind of fill out our database of these different standardized molecular and microstructural data. So again, there's that QR code if you want to look at it more, but we are actually going to dive into more detail about this one. And so I'd love to walk you through kind of how we're involving the scientific community and helping us reach this really ambitious goal. And as we mentioned earlier, EMSL has proposal calls throughout the year that users can submit to. And that really is supporting this user sort of community science. So we have a short form that folks will fill out, and then if they have an approved project, we send them a standardized field sampling kit, as well as a protocol, so that they're able to take those soil cores without having any previous knowledge of soil science or of ecosystem kind of sampling. And that once those cores arrive back at our lab, the Monet team is processing all of those in a standardized way and then providing over 30 different data types. And I'll talk a little bit about that in the next few slides. And that data will also be made available to their team as well as the community as a whole on our data portal. And that can be used by experimentalists, modelers, instructors, and we're really excited to hear from you all of your backgrounds and kind of what type of research you're doing, and if there's some overlap too with some of these decadal goals and where EMSL is going. We've got this fun little video to show what we send to our users, and kind of put yourselves in the shoes of someone who is interested in getting some different data from the environment, from soils, and we send out this kit. It's in this kind of Pelican case that arrives at your university, and then you're able to go out to the field, follow our instructions, and take both in-field measurements as well as the soil cores. And again, we provide all this material. We pay for the shipping. We try to lower that barrier to being able to participate in science. And then the second part of it really is working with our users to make sense of all of this complex data. We realize that these data types are things not everyone has access to at their home institution or wherever they're doing science. And so we've just highlighted a couple of the key data types that we provide to our users. You might recognize, I had mentioned the 21 Tesla FTICR. One of the data types we provide in our suite is actually FTICR data, which again gives thousands of different molecular identities for the molecules that are in this really complex mixture of organic matter in the soil. And then we also pair, we kind of collaborate with the Joint Genome Institute to provide our users with sequencing data so they can know what are the microbes in their soil. And again, we're connecting across what the microbes are doing, what the soil structure looks like, which is the X-ray computed tomography. So we're kind of trying to bridge across these three kind of biogeochemical, molecular, and physical properties to provide that complete picture to our users. And we know there's plenty more data types that could be added, so we're always looking for feedback from our community of what are we missing, what could we be adding or doing differently, so that we're providing even better information to both our users as well as that are in the field collecting samples, as well as our data users. And with that, I'm going to hand things off to Montana because we know that the data is only as useful as the metadata is. And so that's something that we focused on a lot at EMSL, and she will give some more background on that.
I think, Sarah, I don't think that I will have to talk too much to everybody about the importance of metadata and it in data reuse. And if you were at the NMDC session yesterday, you listened to me spiel on about that for like 30 minutes. So basically, I just want to point out that to accomplish the goal of data interoperability and data reusability that Sarah was talking about and AI ML application, we really have to make sure that we've captured the metadata and the important information about what it is you're actually doing, right? Metadata provides the context about anything and everything in science, and complete metadata, it should capture the whole story so somebody doesn't have to leave themselves guessing about what you did. And quite honestly, I've heard somebody call it once a love letter to your future self, because you also might end up guessing what your own information is, right? Like capturing that good metadata at the start will not only benefit you, but also others. And it's at every step and every part of the scientific process. So you saw this figure before from Sarah and also, and from yesterday. It's also at every type of science, right? It's at every type of research, every type of sample. And it's important to keep this in mind so that we can scale from microbes to Earth or going backwards. And again, it's at every step in the scientific process, right? It's not just the research process, but throughout the entire data lifecycle. And it's important to think about it for how you're going to capture your metadata, how you process that data, how you share that data, and how you're going to preserve that data for access in this data lifecycle so that we can continue to go throughout this cycle without having to always start over. And again, for that AI application, for your training data, and for AI use, it needs to be able to understand what it's actually looking at. And we want to make sure that it's not going to lie to us about what it thinks it's looking at. But just capturing that information alone, right, isn't enough. You have to also make sure that, again, a machine can understand it. And when a machine or a human looks at your data in comparison to other data, that it actually knows what it's talking about. And so that's where the importance of leveraging existing standards and ontologies is really important to ensure data harmonization. So we've seen a few XKCD comics in some of my sessions. This one's my favorite, where you have 14 competing standards, and that's ridiculous, and everybody hates that. So we're going to make one that solves everybody's problems. Okay, great. Now we have 15 competing standards. So let's not reinvent the wheel. Like, let's recognize that there are a lot of use cases, a lot of applications, a lot of things that make sense for one community that may not make sense for another, that can be applied in these different areas, and then figure out how we can come together rather than creating something new. And so at EMSL, we are leveraging existing standards, existing ontologies. We're not going to create an EMSL standard. We're not going to create another competing standard. We are going to use what exists. So we work really closely with the Genomic Standards Consortium for capturing sample level metadata. We leverage the Environmental Ontology, or ENVO, FAO, and the Proteomics Standards Initiative for a lot of our ontologies and metadata information, as well as following the guidelines from the IUPAC Gold Book and the Metabolomic Standards Initiative Ontology. And so I wanted to bring this up to show that, if you have a standard that you like, and if you decide that you have, want to write an EMSL user proposal, or you want to talk about science and how we can make sure that science is shared and captured accurately, we can get familiar with those existing standards and ontologies, map to those ontologies. We use a standardized structure for our database that allows us to map to and align with what exists. So even if it's not already in our database, we can add it, or we can connect those fields. And then again, as I said, because we collaborate closely with organizations like the Genomic Standards Consortium, when we learn from the community about what's important or what is missing or what's confusing, we take that feedback back to the existing standards organizations and say, hey, this is what we've been hearing from our users. This is what we did to try and solve this problem. Let's get that into the official standards so that everybody now has this information and this value. And so I'm gonna just ask a quick Slido question. I think, do you want me to ask this one, or? Okay. So you've heard a little bit about EMSL science, and so we want to hear from you. So from this Slido, what data types are of interest for your research and your work, right? So you heard from Sarah about what we do, and so we're curious how this either aligns or what could possibly be missing. eDNA, I'm curious about that one. Who's our eDNA person? Yeah, oh, you too, awesome. I haven't seen a lot, there's Genomic Standards Consortium has, we have a group of people who have expressed interest in like an eDNA extension for capturing that information. So if you're familiar with GSC at all, or if not, be worth checking out. eDNA, metagenomics, 3D molecular, awesome. Metabolomics, curious, is that like LC, GC, or like spatial? Yeah. Nice, awesome. It's okay. I think yesterday we had somebody meant to write soil, and they ended up adding soul, S-O-U-L, so it's fine. So does anybody want to comment on anything for what they found for their, or what they've added for their research and in relation to EMSL? I can throw candy at you too. I forgot to say that. I have candy. Oh, there, see, look at that. The candy is tempting. You can't say no.
Yeah, so I guess I should explain what has just been broken up into a bunch of words here. My name is Ian Nesbitt. I'm with Data One, and a lot of what we do is synthesize metadata standards so that it can be indexed and searchable. We're really interested in sort of how you organize metadata, whether it's on the sample level or the data set level. I imagine that it's probably kind of difficult to organize this on the data set level because there's so many samples, and so each sample probably has to have an ID. And how do you group those into data sets to be able to be more searchable? Because I imagine I imagine Monet is its own search index. So how is that? How are those things organized so that maybe another index can sort of make your data sets available at a broader level?
Yeah, that's so I was at that session. Was that yesterday, day before? I don't know. Time is a blur. But yeah, actually, I thought about that when I was in that session of like, because I do think about it on like the entity level, right, as opposed to the like collection level. And so it took me a little while to like transition my thinking about how we were talking about metadata. And that is one of the things that we are trying to model within our data model, is the ability to say, like, I started here and then it went through a process and get those inputs and outputs that created the path that gets you from sample to data file and then to data set, right? And I think that is part of that connecting with the existing communities of like, okay, when we say that we have this metadata field, if it's called something similar or different, and making sure that we have the right scalability, so that if somebody does want to share to something like DataCite or ESS Dive, if anybody's familiar with that, they have what they need to roll it up and still connect back to wherever it originally existed, and using persistent identifiers to connect to that. So so for the Monet program, every soil core gets an identifier, every individual depth separated sample gets an identifier, and then we're launching a laboratory information management system. I think that's what LIM stands for. That will capture the inputs and outputs, again, that process, the protocols that are followed. But then when you search for that on our data portal, you don't necessarily need all of that information up front to just like look for all of the samples that are from one ecoregion, right? And so we use Elasticsearch to kind of condense all that down and provide a quick way to access that on the data portal. So yeah, that's on our minds too, of how to get that right granularity.
Yeah, I think there are. I think that there are soil chemistry groups elsewhere that have done what you're trying to do with that, and sort of make it into, or have it, have a separate sort of higher level categorization of the cores and the the sections of cores into broader data set organizations.
Yeah.
And yeah, so I definitely think it's possible, and I think it's a worthwhile venture to get that data to be more searchable in other indexes.
Yeah.
And more visible.
Yeah, that would be great.
I just want to put a question out there. This may not be the right time to answer the question, but I'm interested in the fate of the samples that are coming in. Like, are you actually, do you have a repository? Are you preserving the samples? So that would be of some interest to me. But if it's more appropriate to talk about that later, that's fine.
No, I can tell you here now. So The Monet program was launched with the 1,000 Soils pilot project. So over 1,000 data types were generated across 100 sites. So 100 soil cores at least. And now we're in our 3rd or 4th year of Monet, where we've received a lot of soil. We've received a lot of soil, and there were a lot of conversations when we first launched about what do we do with all of this, right? Because we're not using the entirety of the soil core. We use a lot of it. We take 2 cores. One of them goes for like XCT and gets imaging and does hydrology, and then another one gets broken down into its depths and sieved, and that's what we use to extract our DNA and get our natural organic matter via FTICR and total carbon, microbial biomass, right? All of those analyses are done on the other core. But we actually also only do the top and the bottom depths, and so we don't even look at the middle. And we were like, what do we do with all this soil? We got to archive it. We got to keep it all, right? Do we dry it? Do we freeze it? What do we do? Turns out, like, there's just not enough space in the world. So a lot of that ends up going away, unfortunately. We lose a lot of that soil. So as far as like actually archiving the soil itself, I don't think we do that. So we archive the data, and the data is available. But if somebody was like, I would like to look at the 10 to 20 centimeter depth segment, or I want to take some of your soil to do this additional analysis, we couldn't support that because we just got so much substance. I'm gonna add. You can also just come up here, but.
I would just add that Neon is awesome for having soils and being a repository, so we're leaving that to them. We do keep a little bit of air dry, like 2 50 mil tubes worth in case we need to redo something, and we also have, it's a small amount, especially as we've sort of tried to get up and running and expand. That little bit of soil we might even use internally to develop new methods. So we try our best not to hoard soil because, as Montana said, we would be just buried in our lab. But yeah, there was a decision made about not including that middle depth, and part of it was looking at data from the pilot project and seeing that the values of that middle depth largely corresponded to either values at the top or the bottom. So yeah, just sort of make a decision of like, we would rather be able to process more cores and have more data than include that extra part of the core. And we actually do have some cores that the user is not able to take the full 30 centimeter depth because maybe they're sampling in a really rocky location, or it's just younger soil and the profile's not that deep. So that way, we're putting that in the within our laboratory information management system of saying, okay, the 2 depths are actually, it's just 0 to 10 and 10 to 20, because there wasn't a lower section, but we'll still give data for those 2 sections.
You also answered another question I had. So 30 centimeters is the standard depth of the cores.
Yes, sorry, I forgot to mention that. Yeah, we collect, we don't go any deeper, which if you ask a soil scientist, they would say, you need to, there's more carbon there and there's a lot of interesting things happening. We agree. Unfortunately,
So much.
exactly, yeah. And you'd be surprised at how many of our users have a hard time getting even just 0 to 30 because of what I mentioned, rocky, younger soil, compacted, there's so many reasons. And if you get too much deeper, you also run into like needing to get more intensive permits. So that's kind of the other side of things we're not trying to get too far into. So yeah, great question.
Okay, with that, I think we should move on to the next section and turn it over to Kelly Stratton to talk a little bit more about resources and work going on in computing sciences. And then we'll have opportunity for questions in a little bit.
Great, thank you. So like Samantha said, we're going to pivot a little bit, and I'm going to share some more about the work that is being done at EMSL and the capabilities that we can provide for users, particularly those that fall under sort of the computing science area, because as sort of a data-focused week that we've had, we wanted to make sure to be able to cover some of those things in more detail. So in this diagram, we're aiming to represent sort of how the different science areas and research all interact with each other. So we have the biological and environmental sciences represented by those decadal goals of Monet and DigiFin. And sort of in the middle is this computing realm and Midas. And what we're doing there is really trying to build out, and it's ambitious, but build out an ecosystem of AI-based agents that will facilitate data search and discovery, other interactions that we need to have with our data in order to make scientific discoveries. And the longer term vision is that those agents can collaborate both with each other and with domain expert humans in ways that are going to accelerate scientific discovery and especially lead to advanced data integration capabilities. So the computational resources at EMSL are really varied, and some are deployed locally and are frequently used by EMSL staff who are working on user projects. Some exist as web applications and can be used by external users, the public, and internal staff. And then others are accessible on our mid-range scientific computing system, which is one of the resources offered through the user program. And all of these things interact with data sort of across the whole workflow, right? Starting from when data comes off of an instrument or is detected by a sensor, and going all the way through modeling and statistical data analysis. In the modeling space, we work across the scales that were mentioned earlier and have expertise in pore and reactive transport modeling at the ecosystem level, all the way down to computational chemistry modeling at the molecular level. And on the statistical side of things, we have expertise to help with experimental design, data processing and annotation, quality control, statistical analysis, and visualization to really help with that communication aspect and interpretation. And the commonality between all of these capabilities and areas of expertise is that they align with the priorities of the Office of Biological and Environmental Research, which is what funds our user facility. So to help make this maybe a little bit more concrete and get the mental juices flowing more, I'm going to provide some examples of past user research that have been conducted at EMSL, and doing this sort of to help prepare for this Miro board activity where we can hopefully identify some areas of potential synergy between EMSL and the ESIP community and help inform maybe next directions or prioritization for some of our decadal goal steps. So this first example was led by Professor Kalyana Nakshatrala at the University of Houston, and he shares EMSL's research interests in understanding soil structure and function in the context of large-scale ecological processes. And to date, computational challenges have imposed this sort of artificial ceiling that's really limited the community's ability to integrate those pore-scale imaging data into larger scale models. But Professor Nakshatrala saw a way around those challenges, and he proposed a new way to perform that upscaling. And in order to do that, he needed some help from EMSL's environmental and computing researchers to help test that method. So the overall goal is to take this fine-scale soil imaging from XCT images, like you see up here on the top left, and obtain macroscopic soil properties, which is shown at the bottom right, for field or watershed scale science. So the proposal suggested testing the integration of those detailed pore scale simulations, which are very expensive to perform, with approximate but much faster methods that are known as pore network models, and combining those two to perform an AI-based upscaling. So some of the areas where EMSL contributed their expertise to this project were in the XCT experts that we have, which Sarah mentioned, who help to analyze and segment those 3D data from the XCT experiments. Additionally, our scientific computing resources were used to do the transformation of those segmented images into pore network models using existing software libraries. And then calculations were performed using those graph-based models and those traditional expensive simulations with other existing software also performed on the computing cluster. And then finally, EMSL was able to provide additional assistance to make sure that the code, the software, all of that workflow that was built to perform that test was sustainable and reproducible using Docker and other things that we commonly use that might not be go-to necessarily in every researcher's lab. The second research example I'd like to share was led by Professor Lynn Mazzoleni at Michigan Tech and one of her grad students, Amna Ijaz. And they wanted to understand the interactions between aerosols and soils. And to answer this question about what is actually controlling the solubility of atmospheric deposited phosphorus, they needed the ultra-high sensitivity mass spectrometry, which Sarah talked about. That's our FTICR. And that's one of EMSL's strengths, mass spectrometry in general, but also with that really high resolution instrument. So aerosol from wildfires is a major source of phosphorus in soils. And Dr. Mazzoleni really wanted to understand what controls the solubility of that atmospheric phosphorus, because that controls its bioavailability. And the difficulty here is that organophosphorus is really, really dilute in wood smoke aerosol, and it's compositionally complex. So no one had been able to actually determine its chemical form in detail. But using this FTICRMS, which has that really, really high resolution, we were able to characterize the speciation of phosphorus in the wood smoke. And yet Sarah mentioned that there's two of these instruments in the world, the one at EMSL and another one in Florida. And the story that I've heard is that they actually had to take off part of the roof to get this into the lab, because it's the size of a car. It's very large. Anyway, the researchers showed that organophosphorus occurred in two different chemical classes, which are shown by those arrows up here on the right, the purple and the orange. And there's actually about 700 distinct formulae for those phosphorus compounds, which might sound like a lot, but it's 700 out of 10,000. And it was the ultra-high resolution of this instrument that made it possible to resolve those peaks and provide the information that was critical to the understanding and modeling this atmospheric phosphorus behavior in surface waters and soils. I have just three more quick examples. And the thing that I also wanted to highlight is, Sarah was talking about these different instruments and how those analyses are part of the MONET program, but we also use those same instruments in other projects. So we've used those 3D images of soil cores from the XCT to determine pore size, distribution, and the connectivity. And then we've been able to explore the factors that control the organic matter solubility in soils using that. We've also used mass spectrometry-based metabolomics to understand differences in treatments on various types of lignin to help identify targets for characterization. And then we've used NWChem, which is a computational chemistry model on the scientific computing cluster to simulate potassium ion channel gating and demonstrate that protons and water play a significant role in that process. So at this point, I'm happy to pause for questions if there are any, even if it's not about specific science examples, but sort of, how does it work for users to come and do research at EMSL.
Are there limitations on who is allowed to submit proposals for working at EMSL? Like, do you need to have a host institution or be affiliated with any other organization?
I might ask one of the PMs to field that one. You can step up.
Really the only restriction is you do need to have funding to sort of like generate your samples or to fund sort of your own time as a researcher. So a little funky, we don't give money out to folks. And so always a question we get is like, oh, can you pay for me to come visit or pay for me to come do this? And like, unfortunately, no, there are some opportunities. We have summer schools and some other things throughout the year where there might be some funding support, but really the user projects, they're open to really any researcher doing anything if it aligns with what, our mission and what we're trying to accomplish, and again, with our proposal calls. But yeah, we want to work with industry, with any other kind of organizations, academia. Most of our users are from like academic institutions, but that might just be because, that's where a lot of our scientists are coming from who work in the labs. So that's maybe more of our network and community, but that's part of the reason we're here is we want to be expanding that, thinking of new partnerships, bringing in new users, especially in the data space. I think that's something that a lot of us are experimentalists, and so that's kind of our bread and butter. But as Kelly was showing, there's a lot of opportunity for our computing resources and also still bridging across those other science areas. Good question.
So coming back to environmental DNA, I know that it sounds like you're doing a lot with microbes in the soil samples. Are you also looking for other organisms besides the microbes, for eDNA that's telling you about perhaps unexpected other things, even vertebrate DNA that might be present in that top section, for example?
So it's interesting, just anecdotally, the lab staff will find every once in a while, like part of a worm or something else that makes its way into that. And at this point in time, really the focus is that microbial community, and it's with our partnership with the Joint Genome Institute, right? They're providing us a metagenome. And so I know that I think they're working on some different pipelines to kind of expand the data that we're providing our users, but I don't know of any plans right now to do anything in the more of the soil fauna besides the microbial community. But it is something that, I know NEON does a lot of characterization that gets deeper, and we have a collaboration with them where they do our standardized soil sampling and send us cores, and so then we have a link between their sites and all of that really rich data over time with some of our unique data sets that really only we can provide. So we don't do anything like that, but I feel like there are those connections to try to bolster our data and what we provide our users with what they also collect, because it's super important and things that our current infrastructure, I feel like we couldn't support right now.
Yeah, kind of coming in from another planet, it would be super cool if you could kind of collaterally, for example, all the little creepy crawlies that are present in this soil that are playing important roles in what's going on, besides the actual microorganisms.
Yeah, so, more of almost a food web kind of context to things.
I think that would be really cool if you were able to begin to do that at some point, and characterize. This is everything we know about what was present, in that sample, from the tiniest up to the biggest, basically.
Yeah, no, that's a great point. And I knew researchers back in my master's that were really interested in okay, now we also want to know the biomass of all of these different creepy crawlies and get some idea of the, different species in there and the proportion they make up. But yeah, I think at this time we are, because of the molecular focus of EMSL, we're really focused on the microbial community, but maybe there's ways that we can connect to other databases or repositories that have that data and the expertise. I know like you need to have a lot of training and background to even make sense of collecting and using that data. But, yeah, good.
Thanks.
I feel like, I'll also just add that I think that some of the strongest proposals that we see come through EMSL are the ones also that they say like, this is what I do. So like, I care about the bugs, insects, and like large bugs. Can you tell me about the tiny bugs, right? Can you tell me about the microbes, right? And so getting those crosswalks of science, I feel like are usually pretty strong proposals for our facility.
Thanks. That was really great. Can you talk a little bit more about the kind of contextual metadata that you're tagging with the... so it was great to see the list of the different ontologies that you guys are looking at. I'd love to, particularly on the ENVO side and anything adjacent to that, like how are you guys getting concrete about, what you're tagging, what you're indexing, and what kind of problems you faced during that, if you can share any stories as well,
Yeah, so for ENVO, we primarily use that for describing the environment as aligns with MICSS's standard guidance. So the MICSS standard has the environmental, broad, local, and medium scale descriptors for the environment from which you collected your sample. So the biome, something in the general location, like a river, or like maybe there was a fire, right? Oh, and then like actually the exact material that was displaced, what type of soil or what type of, like where the water came from, that kind of stuff. So that is I would say the must-have application within ENVO that we do right now for sample identification. We also have other terms within our metadata that allow for describing chemicals. So like provide the KEBI ID so that we can say that this is the chemical that we're identifying. When we're starting to think about this DigiFen connection and this phenotype link, I had great conversations with Elisha while I was here at this meeting about being able to connect getting from genotype to phenotype and describing that, using ontologies to describe exactly what happened to that organism to get to the phenotype that we see. Again, in a more standardized, structured way so that somebody doesn't have to just say, I heated it up. Okay, well, like here's an ontology where like, here's some things where you apply a heat temperature or something and like describe it, right? So we use it there. We also have looked at like the experimental factor ontology, I think is what that one is called, which is, we've looked at that one for just describing experimental treatments when you so we don't have to just have a string of text.
Is that like, here's the alteration that I did, or here's what this, like, what was it purely an observational experiment, or was there some intervention like that? Is that what?
Yeah, is it a temperature, did you apply temperature treatment? Did you have a moisture treatment? Yeah, what are those experimental factors that are applied to your sample? And then we also just in general trying to, again, link across other standards. So the Metabolomic Standards Initiative organization, it's more of a standard than an ontology, but it's available on ontology lookup service, OLS, of like, you can look up this standard as if it were an ontology, because you get that unique identifier for every term that has a description. So then you can also link to exactly, my standard term within our database when I say that I used, we have a term that it's just substances used. And for us, that means this is, like, I added chloroform to my sample to extract organic matter. Well, okay, they call it something else, right? It's not substances used, it's some other thing. So let's make sure that we're calling it the right thing at the right level. Yeah.
Thank you all. That was awesome. Just to add on to that a little bit, we're also having interesting conversations around the difference between observations and measurements, right? And so that's really why I wanted the microphone. So observations can be very qualitative, right? Like a human sees a green, is a color of green, you know, and then measurements can be very quantitative, right? And so then you need units and you need to make sure that there's a reference to a control, et cetera, et cetera. So those are also parts of what's going into this, how do you describe a phenotype thing?
I was hoping to get more candy, but I guess I only get one. Thank you. So it was interesting, you mentioned the one research project that was began with the actual particles and aerosols to look at the phosphorus. So this question relates to, do you have any people working with water, you know, doing some kind of a sample from water and then applying some of all of the amazing capabilities that you have?
I'm not aware of any. I'm kind of looking at my colleagues here. I know a lot of
Walk up and realize, oh, it's probably actually something I can answer.
aerosols, but yeah.
Yeah, so we do get water samples, and we do also like ice nucleation within aerosols and other stuff like that. And I would say in sediments, right, which have water, we're working on right now pulling in or sharing some data from a user project where they collected, I think it was peat sediment, and there was just a ton of standing water on the sample after it had been like sitting upright for a while. And so they're like, we have a sample where they just like took the water off of that peat and put that through the FTICR, right? Just to see what they get. And then they also did a water extract on the peat itself to get, again, more of that organic matter off. So there are projects that do like look at the water. There's a COMPASS, which I don't know what that stands for at the moment, but it's coastal ecosystem. So they look at sediment and water. The GROW program looks at sediment and water across watersheds. And so short answer, yes. Long answer, I don't know them off the top of my head, but water is of interest.
Sure, fair enough, thanks.
Yeah. Yeah.
So obviously I work with a partner organization to them. So GROW is the Genome Resolved Open Watershed, and they're characterizing river systems. They just had a paper come out in Nature on the US watersheds, and it's a community crowdsourced project, and they sent samples to the Joint Genome Institute for sequencing of the genomes and to EMSL for FTICR. And so they're trying to characterize stream order and the impact of land use on microbial community changes across a bunch of different watersheds in the US. They have global samples that they're processing right now too. It's related to this other WONDERS project, and I don't remember what WONDERS stands for. The acronyms are a little insane. And then COMPASS is terrestrial aquatic interfaces. So they're looking at transition zones in the Great Lakes and the Chesapeake Bay as you get tidal fluctuations or storm inundation and ghost forest development and kind of what's happening with the biogeochemistry. Microbes are kind of like and we'll collect those on the side as much more of a biogeochemical project in general.
Because everybody should care about their microbes.
Kind of to build off of that, has there been much thought about engaging like oceanographic communities? Because they do a lot of sediment coring and water sampling, and like the newer stuff is eDNA. So like what, have you guys put much thought in like maybe engaging those communities and making this like instrumentation available for processing samples?
Yeah. I'm gonna have to give a sad answer to that one, which is that we are funded by DOE and BER doesn't really do the ocean side of things, right? That is almost never in our program calls, sadly. Plug for NMDC, we talked about it yesterday. We can't like come to you and say, hey, give me, but if you wanted to share your data on NMDC, if it's microbial related and it's on the ocean, we could do that. But as far as on the EMSL side goes, unless anybody else wants to provide a cheerier answer, DOE doesn't do oceans. So
It's a lot about how you message it.
That too.
So there are deep sediment, carbon, methane cycling projects that are funded by the Department of Energy because of the carbon cycling stuff or nitrogen cycling stuff. And so like ammonium oxidation or, some of the methanogens, et cetera. So it really depends on how you message it. They're getting more, again, into the terrestrial aquatic interface estuary kind of question. Surface open ocean is pretty much not going to work, but it depends on how you message it.
Yeah. I'll add to that and just say that there's also this, they're looking at critical minerals now too. So that's going to become very important. So if you can bring in critical minerals, then chances are also higher.
Yeah, I was thinking of like air-sea interactions kind of being a newer thing. So like DOE does a lot of like aerosols in the air, and they have sites near oceans that need to be ground truth, and like ships and oceanographic data can provide that data gap. And it's kind of partnering about like air-sea fluctuations, because you only get either the atmosphere side or the ocean side and not the interactions.
Yeah, good messaging. I'd buy it.
I guess I could speak a little bit to like the interactions with like aerosols too. We do partner with the Atmospheric Radiation Measurement User Facility that's also funded by Department of Energy. So we do additional aerosol research with them where they fly campaigns, collect aerosol samples at all of their sites, and then we do molecular analyses at EMSL.
Sure. Okay, next, how convenient that I'm already standing here. We are gonna do an activity in a Miro board. I don't know if there's many people online. There are now 30 Zoom notifications that I feel like weren't there a minute ago, but anyway, we're gonna do a Miro activity, so if you have a computer and you wanna bust it out, that would be cool. And then we can all do this together. So we're gonna just kind of talk a bit about your science questions and where they may fall within those EMSL areas, so biological, environmental, computing sciences, with how they align to EMSL. And we'll share some challenges with that and then brainstorm what we could do if those challenges got eliminated. And so I think that's not the Miro board. I think you can get to the Miro via the Kiko chat. Yeah. And while everybody else figures that out, I'm also going to figure that out. Give me a second. Oops, excuse me. Go away. Close. Fabulous. Oh, look at that. So many people are already here. Awesome. Okay, yeah, so first piece here, wow, scrolls backwards. Create a sticky with your science question and place it on the Venn diagram where you think it might fit best. So there's a collection of stickies over here. Pick your favorite color, plop them in, and drop in some science questions. We've got a couple here that we added. So for example, I study soil parameters from different sites for environmental, or maybe a combo of environmental and computational, being I use image data to extract soil pore network connectivity. Oh, coffee needs to kick in. So yeah, we'll give it a few minutes. Just add some stickies and see what people come up with. And if your sticky moves, it's because we have shuffled them to kind of group them for reasons that will make sense when we move into the next phase. If anybody has any questions, feel free to jump in. And anybody online, hopefully, if you have any questions or issues getting into the Miro, just let us know. Uh-oh, lost it. There we go. All right, I'm gonna fix it. Hold on. It's okay. These were locked in. I don't know what happened. I'll fix it. We love Giro.
For people online, the question was, do projects that fall in the middle have the highest chance of being funded by EMSL? I think that for science in general, it is great to be integrating as much as you can, but I don't think we could explicitly say you have a higher chance of being funded.
Not always.
Yeah, sometimes the projects will be focused on really just one of the science areas, and that's fine. I'd say one thing we tell proposers is think of like how best to use the resources. It's really easy to say, I want to use 10. I want to use everything. But if there's not a clear, use for all those different data types, the peer review panel is going to see that and say, okay, you just want to touch everything. That's cool, but like we really want to further, these scientific areas and be able to add value to the research that's going on. So I really think like there could be a project or proposal that has 2 techniques, but if they're used super well and integrated, that could have more scientific impact than one that wants to use 12. So I think it's tough because it is, you see all the shiny toys and you're like, ooh, I want this and that and other. And we encourage proposers to work with our scientific staff because they have that expertise. It's really helpful to make sense of, okay, I have this question, what would be good techniques to use? Because something might look good if you just read through the website, but talking with a scientist will help make more sense of like, well, what actually will you get? Will it help answer your question? Do you need qualitative, more quantitative data? There's a lot of nuance. And so, yeah, it's not always the one that requests the most that's going to score the highest by our peer review panel.
Yeah, the other thing that I wanted to add is that, it's important too when someone is proposing work that they're clear about which parts they want done at EMSL versus what they can do, with their own research team, right? So there are some projects where maybe they haven't worked with a certain data type, so they want help with the analysis and integrating that with other data. Or maybe it's a type of data that they work with routinely and they're totally, experienced. They know what to do, they know how to run a model that they want to apply, and so they don't need help from that computational analysis side. They need the data from the instrumentation. So it's a mix.
Yeah.
Okay, I think I've seen stickies slow down, so I'm going to drop a little square to just kind of give you a sense of where the next step might align. So if you added a sticky to, any of the areas where my little squares are popping up, if you scroll to the right, there is another section here with what are challenges associated with these science questions. So thinking about, the science question sticky that you put in, and whichever area of these little green, blue, or orange boxes, again, drop another sticky and add some, I'm sorry if I'm getting everybody like motion sickness from moving around on this main screen so much. It like looks very different here than it does there. And I'm not used to not Max. Add a sticky note with what are some challenges associated with that science question that you have. And if you have a hard time moving around, or if the other cursors are bothering you, I should have said this at the beginning. If you don't know, you can click this button up here at the top, and you can tell everybody else to go away. And you can also click on a name and it will jump you straight to that person. So this is not me. There we go. Now if you click on my name, it will take you right to the correct spot.
Really curious about this green sticky and that orange square that says challenges analyzing complex data sets. I'm wondering if someone can elaborate a little or give a couple of examples about the types of challenges.
And data sets.
Yes, and data sets. I don't know who wrote that one.
Yeah, so I wrote that one. I think with this rise of big data, thank you so much. I think it's, at least for me, as someone who's not really a data scientist or someone familiar, I'm okay doing things on a single genome scale of assembling genomes and things where I feel like I have a good handle on it. But I think there's been this rise in wanting more and more data and then more integrated into one science question. So publicly available data, my own data, and so linking across data types. So I feel okay with genomic and transcriptomic information. I feel very not okay with metabolomic data. So figuring out linking between things like genomic information and functional information is really hard for me, and making the connections with data types that I'm not familiar with and figuring out how to best analyze those, even on a small scale for those, but then for everything as we're kind of scaling up in amounts of data, and then how to integrate that and kind of pull together more data types, more numbers of data and things like that. That's something that I wasn't trained how to do, so it's kind of learning on the fly and figuring out how things work and yeah, how to work with that stuff.
Thank you. Yeah, I think that's definitely a challenge across the board. And it's exciting for me, coming from a background in statistics, of like, how do you integrate data that's different scales, different either on time or like relative size of measurements. So there are ongoing research in that area, and then also, depending on the types of data, some established methods out there.
I think that was, okay, so yeah, so we'll do a little more discussion now since Kelly kind of kicked us off. So we've got this, I think you can maybe read that. Need to know soil composition in addition to biome behavior. Does somebody want to talk a bit about maybe the science question with that, and a bit about the challenges they face?
Yeah, I wrote that because my previous research when I was in academia was looking at watersheds sort of in totality, more from a sedimentary perspective, but wanting to know what kinds of things were influencing the watershed from an environmental and biological perspective as well. And so the science question that I wrote was, how are microbiomes in a given watershed consuming resources? And so, needing to know what they have available to consume, and how they're, how they particularly use those resources was, something I decided to put in this square. So.
Yeah, I think that's a really good question, a really good point too, especially thinking about from like you said, sediments or water ecosystems, because it just kind of flows right in. So getting that connection and integration of like, maybe there's a farm upstream where a lot of cows, and you have no idea that that's what's causing the response that you're seeing. So that data integration and just understanding the ecosystem around is really important. Yeah.
Yeah, for me specifically, it was geology related, and so I was thinking a lot about the composition of rock types in the areas and what that was able to support in terms of microbiomes.
Right, yeah. Oh my God, scrolling so backwards for me. All right, no boxes in the blue so far, but we've got a good number in the yellow or orange, which I think was data related, if I remember correctly. So anybody want to talk about their challenge? Or does anybody see a challenge up here that they're like, oh, I hadn't thought about that, that's a good one, and want to talk a bit about their science question? Multitude of standards with little conformity. Yep, preach. Me too. Yeah.
Yeah, I'd love to hear more the challenges with making the right team with wet lab and computational experience. Like, not sure who wrote that, but just to understand what do people see as like the computational and software expertise needs in the kind of either what you've written or, what other folks are thinking about.
Yeah, I wrote that. I think we do see this a lot of, we have to almost become non-experts in a lot of things too. So I think there's been a rise of people understanding the full process and having the wet lab and the computational skills, but sometimes we really like having access to someone that is the expert in this area that can do the best job analyzing that data versus me taking 3 times as long to do a worse job at it. So I think figuring out how to make a team composition and then work together and kind of speak the same language. If it's a fully wet lab person speaking to a fully computational person, there's sometimes that gap in why didn't you do this many replicates, or is it not feasible to do this at a field scale, things like that. I think we've struggled with the translations between having fully computational and fully wet lab, and then being in the middle, not being like an expert of none, and kind of still improving your skills while also trying to figure out the team composition that would be best suited to put things together. I think we've definitely just struggled with that a little bit.
Yeah, Elisha left, but she hosted, I think with you, a DM data management plan tool session that I think one of the things I took away from that was you have to think about the end piece at the same time as you're thinking about how you're designing your experiment, and transitioning the narrative of, the research life cycle to do that early. I don't know what the right answer is to get, people to do that, but yeah.
Yeah, I also see a lot of overlap between this issue and the purple one about little conformity, because I think part of the issue is like the model that a lot of university PIs operate under is if they're really lucky and have a lot of money, they get to hire their own, data person or computational person. But that person is typically overwhelmed, often is self-trained, meaning maybe started in the wet lab and then like developed computational skills. And so their kind of motivational structure is just to serve the like 5 or 6 postdocs and grad students they work with. And maybe they don't even know. I mean, I have known such people that aren't even really aware of like global ontologies, just because they aren't supported by the infrastructure that, folks like working in a national lab necessarily are. So I don't have like the direct solution to that, but in some ways, like I know in the biomedical field, they've moved towards more centralized resourcing, meaning let's say you are a doctor or like an MD, PhD researcher at a hospital, instead of having to go out and hire your own biostatistician, the hospital has like a team of biostatisticians who are all talking to one another, and it's like flat rate written into your grant to work with them. And I think that kind of helps towards the conformity piece.
All right, there really should be clocks in this room, because we got 6 minutes. So last piece of this Miro board, again, you can follow the little lines. Oh my God, this is so hard. What would be possible if this science challenge for your research question were solved? What could you do? What could you answer? What could you solve? I hope somebody writes world peace and climate change, but here we are. Solve all the problems, fix all of the world problems. But yeah, and also if anybody just wants to speak to what they feel like they could do if they could solve that science problem. More time for research. Is that assuming that AI can help us fix everything? Because I think that would be great. That's something that I've thought about a lot, listening to a lot of the sessions here at this conference is, we have to make sure that we get AI right. So we don't end up with a bunch of garbage in, garbage out, crappy science. But if we can get it right, then I think it does have the opportunity to free up a lot of very intelligent minds to create a lot of thoughts.
I'm feeling left out and want a piece of candy. But K-12, I'm just going to throw a little pitch out, grow your data scientists in K-12. I went to a K-12 data science education in San Antonio this last spring, and there's a whole bunch of us that are trying to figure out what do we need to do down there so that all this can be easier at this level.
Yeah, I love that. And we think about that a lot of just like educating and getting people to understand what to do to start. We spend a lot of time with our users at Emsl, just like on one-on-one calls to be like, let me explain to you and like, let's help you understand this data and let me help you understand this metadata and what we're trying to capture. So that way, when they do it again later, it's good to go. We have an NMDC ambassador who had a user project. He's an NMDC ambassador, and he submitted his metadata to Emsl, and I was like, bro, what you doing? This is terrible. Like, it was awful. Like, it was not formatted well. Not all of the samples were there. There was information missing. So like, we sat down, we had a conversation, he understood, and now it's like his metadata, he sent like 4 or 5 more sample sets to Emsl, and every time it's like, good to go, ready to, all set. Like, helping the community grow and learn, yeah.
Well, just to follow up on Carla, are you getting proposals coming from K through 12 institutions at all?
At Emsl? No. I don't think so. That's not a, yeah. So far, I feel like with Monet, that might be the easiest route to engage, because there's a lot of things on here, a lot of techniques that would be really difficult, I think, for K through 12 to like produce, an experimental design that's competitive against, R1 institutions. That's tough. But I think Monet having that standardized kit,
Right.
We want to provide the material so that anyone can do it. The form is really simple to submit to then participate in it. I would say we have proposal calls that come out on Emsl's website. And so if you're interested, we have a newsletter you can sign up for. So just like to keep, that in the back of your mind, because I think we would love to work more with K through 12 and like help you figure out like how would you sample, what are the needs. We do need an access agreement. So like if you're sampling on someone's land, taking those cores, we need to know that like they've given explicit acknowledgement that that's okay to do those activities. But really besides that, there's not anything for like, we're not putting any other requirements on our users for Monet because we don't want it to be a barrier. So that is an area we want to get more community engagement.
The problem is, I think a lot of us are stuck in our kind of academic, like academia-centered social circles and professional circles. So I love that you're here and that you came to our session, because I'm like, it would be great to have more engagement too, especially with like having students go out and collect soil cores, and then we could have training sessions of like, how do you make sense of, soil pH and the like microbial biomass, and like what do those measurements mean? And our data portal, as I said, is open too. So that's another way to get involved if it's not through submitting cores, still working with some of that data. And we do have an inbox. It's monet@pnnl.gov. And so, yeah, I would encourage you to like send an email. I'd love to connect about this and see if we could even just do like an informal sort of like training session and go through a couple data types using our data portal and just like see how students interact with it.
I could picture just dynamite science fair projects.
Yes.
Coming out of this, and so to like kind of seed that and start to build some momentum,
Yeah.
so many, at least at the high school level, I think I could just really visualize cool science fairs. So yeah.
I 100% agree, yeah.
I know we're at time, but really quick, this is making me think about, we have all of these data that have already been generated, that are available through the portal. So even if it's not writing a proposal, there are existing data sets out there.
Yeah, I did. I've already signed up to run this thing. I got into your portal, and then I thought, is this free? Is it okay to download? Yeah.
you can
Once I
download.
logged in and created an account.
Yes.
You won't be charged for any, it's all free.
Yep. So yeah, happy to talk more about that too, one-on-one.
All right, do you want to give the closing sign? Yeah.
Okay, so thank you, everyone. This was a really great session. I really appreciate all of the interaction and engagement, especially with something that was, I think, new to all of you. So up on the screen, just have a few ways you can stay connected with Emsl. You can email us, you can follow us on social media at Emsl Science. The QR codes will take you to sign up for our newsletter or look at the website about open proposal calls. And then for those that are in the room, we do have a few handouts if you're interested, just a few more details about Emsl's science, some details about the specific data types that are collected as part of Monet, and then a little pamphlet about our automation technologies and where we're moving with high throughput science. So thank you.